can't believe the Porphysome has been 10 years old. And so I like to, we have a lot of experience, good and bad. So I like to share our experience and maybe some lessons we learned from this pathway to the clinical translations. So one of the key nanoparticles hallmark is really multifunctionalities. And by the same token, theranostic, which is a field which is covering both therapy and diagnostic, is also within the realm of the multifunctionality of nanomedicines. So as a nanoparticle goes, you can really load different drugs, drug combinations, imaging agent, and in terms of theranostics, you can actually have imaging combined with therapy has offer a lot of options, from imaging drug delivery to stratify patient for find the right population for using for the therapeutics. You can also using using non-invasive way to imaging the lesion, then followed by the other guided therapeutics. So usually there is a two method to making nanoparticles multifunctional or making theranostic. One is the most common approach, which is we consider like a Lego approach. You combine different building blocks, which each one has one function. Put them all together, it's called all-in-one approach. Now all of a sudden, this nanoparticle can perform so many different functions that way. And this is actually very useful and has been already in the clinical translation, clinical use in a way, because there is many multiple components readily available from a clinical approved agent. When you combine together, this is actually help the translation in a way you can actually also build in the different functions. But the challenge is when you are going beyond actually two modalities, the particle become very complex. And this creating a lot of issue in terms of reproducibility and the toxicology issues in terms of in vivo, you have to characterize the in vivo fate, because FDA will require everything injected in the body has to be counted for. When you have one or two starting to become beyond two components, then this become very complex scenarios. Another approach is what we actually been focused on is to think if you have a building block molecule for nanoparticle, it's multifunctional, then you can build the particle with this multifunctional building block. Is it contain all these functions, and that will actually help to the translation pathway, because in theory, all you need to characterize that building block to make sure they can be reproducible, and make sure they can be tracking their fate in vivo. Now you actually can apply that to different applications without going through every single clinical translation hurdles. But the challenge, obviously, is very difficult to find these inherently multifunctional building blocks. So our lab is very interesting in this porphyrin molecule, which is a naturally existing theranostic building block. For example, the porphyrin, the iron porphyrin is existing in the blood, which is in the presence of heme. In the green porphyrin is a magnesium chlorophyll. Porphyrin is existing in the chlorophyll molecule. But as porphyrin in general is a very, very well-known biphotonic agent for the imaging and therapy in terms of fluorescence imaging and photodynamic therapy, where the porphyrin interact with oxygen and the light to generate reactive oxygen species, mostly single oxygen, to leading to the cancer kills. But the metal chelation property of the porphyrin can also be taken advantage of, for example, by replacing the magnesium in the chlorophyll or the iron in the hemoglobin with a radioisotope like copper-64. Now you transform a photonic material into a nuclear methane for PET imaging tracers. In fact, our lab has been working on many years, trying to take advantage of this multifunctionality of the porphyrin to do exactly that. For example, in this example, we have a PSMA-targeted porphyrin agent, and which is showing up by PET imaging, showing up the very specific PSMA positive imaging. And the same can be shown by the fluorescence imaging, and we can lead to very selective cure of the tumors by this photodynamic therapy, by the PSMA targeting. But in order to make them nanoparticle is actually not that simple. So 10 years ago, my student John in the lab came up with an accidentally discovery. What he found out, when a porphyrin replacing one of the fatty acid chain of the phospholipid, this molecule can self-assemble into a liposome-like structure. Because it's a porphyrin, because it's a liposome, so we call porphysomes. It has a very thick biolayer, which reflecting the porphyrin in the biolayer structures. The discovery of this one is very interesting because you think about it, when you porphyrin a dye absorbing light, when you have 100,000 of them come together, it's like a black hole. You're absorbing all the light you can get, but nothing comes out because everything self-quenched. There's no fluorescence, there's no emitting photons. It's like a black hole because the light gets in, the only thing comes out by heat. Take advantage of phenomenon, we were able to show photothermal therapy can be very efficiently performed by injecting into a mice with intravenously with a porphyrine nanoparticle. When it's accumulating the tumor, you shine the light. Now actually you can see the temperature rising very rapidly over the 60 degree, then leading to tumor necrosis and within 1 minute. And that leads to a very efficient photothermal ablation agent. You can also doing the photoacoustic imaging, which is also a thermal behavior. The only difference is instead of continuous wavelength laser, you are using the pulse laser, which means you generate only a transient temperature increase, lead to the thermal expansion detected by acoustic wave by ultrasound. That means lights in, sound out. It will give you a very strong photo, very high resolution image in the deep tissues. For example, here, you can see the lymph node vessel can be clearly, lymph structure can be clearly delineated by the porphysomes, while before the injection, you only see the vessel, which is from endogenous porphyrin, the hemoglobins. Another thing is, when is a porphysome structure is a nanoparticle, everything's quenched. But once reverting back to the porphyrin-lipid monomers, the porphysome enables a low background or activatable fluorescence imaging, because this time what you have is the fluorescence imaging have been activated when it's porphysome into the tumor, in the tumor areas, which means you have a selectivity. So for example, we can do the lymph node dissection using the fluorescence imaging by the porphysome. You can see the lymph node is clearly, can be delineated. And after lymph node dissection, you will see the residual tumor, residual metastatic lymph nodes still be shining up, still be lighting up, and that will lead you to complete resections. You can also do photodynamic therapy because a porphyrin is a very good photodynamic therapy agent. When it's revert back to the monomer, it gives you a very strong photodynamic activity, and is also be much more selective because of the porphysomes accumulating tumors. Another important point, as I mentioned, porphyrin can chelating metal. What we found out, the porphysome by themselves can chelating metal very easily. In fact, very robust. You get about more than 90% labeling yield, about incubate all you need 30 minutes at 60 degree. The radio label gets in. After 30 hours IV injection, the copper is still remain within the porphyrin structure, which means what you see is what you get, because your nanoparticle itself is a chelator, so you don't need to add more chelators. And that means the porphysomes in vivo fate can be tracked by these PET imaging tools. But because the PET is very sensitive, if you want to chelating maximize, every single porphyrin can be chelating with paramagnetic metal, like manganese. Now all of a sudden, you have 100,000 MI contrast agent. Now you have a very nice MI sensitivity for the MI contrast. So you can also load the drugs, just like liposome does, because porphysome is named after liposome. You can target it. We can actually input antibody targeting, a small molecule targeting, or peptide targeting, just like any nanoparticle do. The bottom line, a single porphyrin-lipid molecule building block self-assemble into a porphysome structures to providing all these functions. So this is what this one for all concept has been popularized. So over the years, we actually have validating the porphysomes preferential tumor accumulation in many, many different animal model preclinically. In the cancer type, we have done head, neck, prostate, lung, pancreas, brain, thyroid, ovarian, endometrial, sarcoma, many different disease. Haven't tried the breast yet. Everything else has been covered. We tried different tumor model with subcutaneous orthotopic, patient-derived xenograft, immunocompetent, chemical-induced, and metastasis models. For the species, the lab actually creating the moon model, for example, from the mouse, rat, hamster, rabbit, dog, and we even have a pig tumors, which is a pseudo-tumor because they don't grow tumor easily. And I want to give you one example on the porphysomes multifunctionality and using one single animal model. This is the Bchl, which is rabbit oral cancer tumors. We actually seeded also topically into the rabbit oral cavity. What you see is by pre-operating using the essentially the PET imaging, we can see a very clear delineation of a tumor, like a fireball here. And you actually can see the drainage to the lymph node, which is metastatic. And our clinicians collaborate very excited about this because if you can see that in the pre-operating setting, what if you see that in intraoperatively? Indeed, in the intraoperative setting, we can see the same pattern reveal itself. We can see the fluorescence when the skin is intact. You can see the fuzzy because of the scattering. But once the skin flap is removed, you can see the tumor was revealed themselves in the tumor bed. After the tumor completely removed, you actually can see deeper. You can see the lymph node, and you can see from the first lymph node migration to the secondary lymph node. So the whole chain you can actually observe from the primary tumor to the first metastatic lymph node to the secondary lymph node. You can all be removed. So it's a very powerful technique. The same system we can using MRI to see if using MRI we can see the delineation of the lymph drainage as well. In the meantime, we can actually do the photo treatment because the agent is already there. So you don't need adding another agent. You just shine the light. You can do either by photodynamic therapy, you can do the photothermal therapy, can lead to the tumor ablations. So that's just an example how the multifunctionality is demonstrated. Now the key question is how we can move to the clinical translation. One way to do it is getting a company support, getting industry sponsors, and everything you just, hands off, let them take cell. But unfortunately, we don't have industry support. But this is too good to not pushing for the clinical translation. So we really embarked almost like 10 years journey to push this translation. And a few lessons we learned. First of all, we have to establish the impactful clinical scenario while taking advantage of the porphysome's intrinsic multifunctionality. And second, we also have to address the critical manufacturing, safety, and regulation challenge that giving the porphysome status as a first-in-class agent. So it's not like antibody, you can radio-label them because antibody is already clinical proof, and radio-label them will be much more easier. However, this is the single, first a new chemical entity you have to go through as a drug, not as imaging agent. So to explore the clinical scenario, I want to give you one example, the first inpatient scenario. This is in early stage lung cancer, peripheral lung cancers. And in this kind of scenarios, we will have a patient, ineligible for the, we have a lot of diagnosis of cancers, of the lung cancers, especially the nationwide screening, find a lot of cancers. Most of them are actually not aggressive, right? But you have to do something. You have to, normally what you do is to do surgical removal of partial lung, or you can do, currently people trying to do SBRT is as another way to address this. But really the challenge is when you see so many of these lung cancer patients, and not all of them are not only a surgical candidate, but it's also not unnecessary to do a surgery. So we want to find out an optimal treatment which is high efficacy and high specificity. So to prove this, to work on this, we develop a scenario that is through the transbronchial way, which give you the very invasive, non-invasively. Through the bronchoscope, usually people do the bronchoscope to the biopsy. But if we go through the bronchoscope, we feed in the fiber through this bronchoscope channel, we can actually detect the fluorescence imaging of the tumor shining by the lighting up by the porphysome fluorescence. Then we can stick the fiber to ablate them directly. So we can avoid all this, only just doing biopsy, we can actually treat the tumor in situ. And to do this, we actually have to build a model, which is our lung cancer model in the rabbit, because rabbit is about 4 kilograms, like a baby. And we have access to the smallest bronchoscope, pediatric bronchoscope from Olympus. So we were able to use a rabbit seeding the lung, tumor in the lung, and showing the very same pathology as a tumor encroaching from the bronchial site. We can observe the tumor. Now we actually put into the use, inject the porphysome, 45 minutes later, deep in the bronchial, through the bronchoscope, we didn't observe anything. But 40 hours later, as the porphysome starting to accumulate in the tumor, and then when they're starting to delineate themselves from the tumor, from the background tissue, because a lot of them are washed out from background tissue, the tumor actually revealed itself behind the wall. You will see 1 and 2. If you look carefully, there's a 2 tumor lesion is shining behind the wall of the bronchial. And the moving fast is because rabbit is breathing much faster than human. But this really underscores, if I don't see the tumor behind the wall, how can I insert a fiber? How can I do ablation? And this is how we did it, because we see the tumor, we do the fiber insertion, and ablate the tumors. So this is a first demonstration of the concept to the imaging and fluorescence-guided ablation of the tumors. Then we want to prove this is actually biological relevant, because a VX2 tumor is a rabbit tumor, it's not a human cancer. So our really amazing surgeon collaborator was able to, in the Kazuo Yasufuku's lab, was able to actually getting this beautiful model done without doing surgery, essentially through the oral gavage. You see the vocal cord, getting the tumor seeded into the lung, then this creating a very different cell line model from A549, H460, H520, all 3 different kinds of non-small cell lung cancer. We're able to show the tumor selectively taking up the porphysome compared to the normal lung. So validating the biological relevance. Then we want to prove the clinical technological relevance, because the closest to human lung are the pig lung. So, but pig doesn't grow tumor easily. So what we did is a little bit step better. That is, we're creating the tumor in the mouse, inject the porphysome, get accumulated into the tumor of the mice, then taking the tumor out, seed it directly into different location of the pig. So this is not a pseudo-tumor, but is actually better than the matrigel. So we actually have a distribution of the porphysome in the tumor. This way, we were able to demonstrate the transbronchial fluorescence imaging, transbronchial fluorescence-guided biopsy, and also the transbronchial photosome ablation, all being validated, showing the technical feasibility of this one to do this treatment protocols. But that's not enough, because we recognize the bronchoscope has only reaching to a certain area, cannot reach the entire lung, because of as you go deeper and deeper, the lung become bronchial, they become smaller and smaller. So in order to do that, we actually think about one way to do it. As the bronchoscope cannot go deeper, the fiber can, right? So in collaboration with a Japanese fiber company, we were able to demonstrate creating this fiber scope. And after you reaching to the bronchial, right? Now insert the fiber going further. This fiber is 1 millimeter outer diameters, contain 2 channels, one for imaging channel, which you can acquire the fluorescent imaging of the tumor. Second is ablation channel with the phototherapy laser. And that is very exciting, because this 1 millimeter outer diameter fiber can give us a 6 centimeter deeper of this penetration in the lung. That means, in theory, we now can cover the entire lung areas. So this will, only when you have cover entire lung area, this become a practical to do this imaging, minimally invasive imaging-guided phototherapy ablation way. So once we prove this concept, now we actually want a device working with it. And this is the prototype device we came up with. It's still too expensive for the application, but this is almost like a Ferrari, right? I always say it's like a Ferrari because now you can do fluorescence imaging, you can do photothermal ablation, you can do photodynamic therapy, all into the one equipment. So now we're working on finding a way to get a much simpler, much cheaper version so we actually can be put into clinical use. So the next, one thing is about this find this impact scenario. We actually have a couple of other impact clinical scenario in the work. Another one point is we also think about the first in human strategy is we actually, in order to take advantage of the intrinsic porphysomes, intrinsic multifunctionality, we actually worked with this phase 1A, 1B hybrid design. So the idea is as follows. Because of porphysome is a first-in-class agent and has to be treated extremely as a cautious for the safety purpose, we have to start with very low starting dose. The best way to start low dose is a PET imaging through the copper 64 porphysomes. And we will do that with the metastatic advanced gynecological cancer patients. And this will give us the first feature, whether the injected nanomedicine is safe, right? Whether because the PET imaging also gave us a chance to do the imaging of pharmacokinetics by distribution, and even for the imaging feasibility as a staging tool, because you have to providing benefit to these patients in order to do the first in human trials. Once the safety is confirmed, we can start simultaneously at multiple sites. In our plan, we will try with the lung, oral, prostate, and gynae, and all at the same time. Now we can explore the therapeutic potential, whether it's for fluorescence-guided surgery or for the imaging-guided photothermal ablation or for the photodynamic therapy. This will collect all the safety data again for the mostly on safety, but it will actually for design the future therapeutic trials. So we obviously encounters numerous challenges toward this first in human porphysome strategy. I want to show a few examples. First, I'd like to actually acknowledge Michael Valek, my brilliant student, who's been driving this, to overcome all these hurdles together with many of my clinical partners. So the first is isomer challenge. As a chemist, we were very happy to find out as a porphyrin phospholipid, because the phospholipid has a two radioisomers, SN1, SN2. As a chemist, you love it because this gave you a completely two different isomers. We actually observe there is no difference in their function. However, we observe in the in vivo system, they actually degrade different way. Their biodegradation pathway are different. The speed of degradation are different. For example, in this SN2 isomers, we observe, you will see in the serum in the blood, they remain intact. But in the liver and the spleen, they actually see majority of them are starting to break down. This, as a chemist, very happy. But when you go to talk to Health Canada, they say this is not good because you have to either give me a single isomers or you give me a fixed ratio of radioisomers before we actually allow you to go even further, because everything has to be trackable and in vivo fate has to be determined. If you have two different in vivo fate, that's a red light. So this means we have to come up with a way to do the study of the metabolism study for each isomer. We also have to figure out a way to scale up manufacturing of a single isomer as a building block. And that takes us a couple of years. We were able to demonstrate metabolic characterization of all at both isomers. Most importantly, we're able to achieve the 200 gram scale of these porphyrin monolayer, lipid monolayers with a high single isomer purity and a yield. And this is a very challenge. We have spent a couple of years working on that. Once we do that, we actually want to work with because we have to do in-house, we have to work on the PET imaging, we have to do the radiopharmaceutical kit development for single patient dose vial in phase 1 study. And for that, we were able to design a single dosing patient vial for 635 milligram porphysome in 4 ml. And we did all the release testing in terms of GMP condition, all in-house. And we're very, very excited and thankful for the NMIN Pharmacol and the BCCA, Dr. Marcel Bailey, who's now taking up our work to do in this clean room production of the porphysome, which is already started. So very excited about that. And thanks for Dr. Bailey for that. We also, now we have this patient vial design, how we're going to radio label and how we're going to study the different dose escalations, right? So our idea is start with the single vial. We can have half of the bottle is half filled. You have basically a buffer, sodium acetate buffer is also clinic GMP, patient use. Now you having this buffer with the copper 64 and inject into this vial with a porphysome, sitting for 60 minutes, now you have injection to the patient. So this is a radio-labeled one. But by changing the dose, we will not change the radioisotope dose. We will radioactivity. We will actually mix with the unlabeled porphysome. This gave us many different dose escalation with the same radioactive dose. So this gave us the chance to see from the PET imaging dose, we actually can go to the phototherapeutic dose. But safety challenge as a first-in agent is very, very tricky and very rigorous. So we actually confirm, validate the neural studies in the 4 species, in the rat, rabbits, beagle dogs, and non-human primates, all in by ourselves, especially in the beagle dogs, right? We did a 40 in-house in our building. But the first dog, we actually encountered some infusion-related reaction, and that is related to the Bchl-lipid. At that time, we have no idea about. Eventually, it was resolved with changing the infusion protocol, so we were able to navigate that. But because initially we observed some of these infusion reactions, which are common for the lipid nanoparticles, at that time we have no knowledge of, were asked by our clinical partner, we had to actually validate in non-human primate. So I have no choice but to send all my 2 students, Alice Carr, to China to actually do these monkey studies. And that was fortunate, that was before the pandemic, otherwise we can't do that, right? But in this monkey study, we observed no infusion-related reaction, no behavior or weight changes observed, no histopathological changes. Everything was perfect. There's not a thing goes wrong. So this reminds me, perhaps in the future, for when you people dealing with a lipid nanoparticle with a pegylation, skip the dog, go to the monkey directly. It will be my recommendations. That's a lesson we learned. Because we're working on the photo agent, so we also have to do some dealing extra work on the photosensitivity study. For that, we actually did a very thorough study to compare the skin photosensitivity. We observed within about 8 days, we will actually have no skin photosensitivity after injection, while the clinical control has a very high photosensitivity. So we were very happy about that. And finally, on the clinical front, we also now starting the clinical trial with the Ontario Veterinary College in Guelph University. The first patient is already done with the thyroid cancer. The idea is because of not only we are treating this veterinary patient, but the dog tumors are naturally occurring, which give us a lot of indication of a human tumor example as well. Particularly, we're actually interested in the oral cancer cavity and the thyroid. This is a canine tumor, this is a human tumor. So this gives us a way to validating the agent safety and the phototherapy trial safety. So this will give us a lot of indication how we help the design the clinical trials. So our goal is first in human this autumn. I've been talking about this almost like crying wolf. I'm talking about this every year, but now I think given everything we've done, this is quite realistic. So before now I want to shift gear a little bit, because in addition to the porphysome clinical translation path, we also are working on many different nanoparticles and beyond porphysome, really creating a family of the porphyrin nanomedicine or microparticle with different size and shape and composition and different functions. So I'll give you 3 examples here today. So the first example is, before I talk about that, is when we changing the how the porphyrin-lipid how they self-assemble. Only using the same building block, we're able to getting very, very small, 10 to 20 nanometer, the HDL-like particles. We're able to getting J-porphysomes, porphyrin nanoemulsions, porphyrin microbubble, a giant porphyrin vesicle as serving as a artificial cells. But if we changing these porphyrin structures by removing that with a different porphyrin, we will get a nanotoxic, or even a non-porphyrin by Bchl-lipid, we got a Bchl-lipid. And we're now actually working a lot on using this porphyrin lipoprotein, porphyrin lipid nanoparticle for the RNA controlled RNA release. So the first example is porphyrin microbubble. So why we're interested in that? Because ultrasound microbubble has been widely used clinically for improving the vascular imaging. Last decade, you also see a huge change in terms of the field is getting very excited to using this beyond imaging, using the therapeutic potential of this microbubble with ultrasound. For example, the lipid microbubble, 1 to 6 micron, they actually can induce, but through the stable cavitation, initial cavitation, they actually can induce many different biological response. And in fact, in Sunnybrook, my colleague, Carlo Hanning, were able to actually open up blood-brain barrier and for the different cancer and imaging and for the different disease applications. And this is where we're excited about the microbubble. So combined with the porphyrin phospholipid, we were able to demonstrate the porphyrin lipid shell microbubble can be formed with multifunction, including the ultrasound function, photoacoustic, and fluorescence imaging. Because the regular microbubble only has ultrasound function. Once the bubble bursted, there will be no more signals. But in this case, we actually can observe the photoacoustic and the fluorescence at a different stage of these porphyrin aggregations. This allows us to be able to determine once the porphyrin microbubble, when you irradiate with ultrasound, we actually observe that the bubble, the big microbubble becomes a nanoparticles. And these nanoparticles can be retained in the tumor for a very long time. That means, for example, here, without the ultrasound bursting, this microbubble with the porphyrin microbubble will be actually photoacoustic signal will be disappear very quickly. But if you add in the ultrasound irradiation in the tumor, 60% of photoacoustic signal of this porphyrin observed over 2 hours. Really, this means you can actually have nanoparticle accumulation in the tumor can be dramatically increased and very instantly by using this ultrasound irradiating technique without going through the EPR, right? So this is actually potentially suggest a new EPR independent active delivery nanomedicine. But more importantly, we actually hope this porphyrin microbubble, when they converting this nanoparticle, they contain gas, they are nanobubbles. But to do that, Carly, my student, came up with this brilliant approach to design a technique. In addition to this ring ultrasound transducer for the transducer, she also adding a receiver at the window chamber. So that means simultaneously, we can monitor both the fluorescence and the acoustic behavior of this, this conversion process. So in the solution, we observe this is the most likely this daughter nanoparticle, a nanobubble, because we observe this TM shape looks like a bubble. We also observe these bubble actually has a buoyancy. So you actually know they are the bubble. But in vivo, we can now confirm that because we're able to demonstrate extravasation of this microbubble after ultrasound bursting, leading to 5 times more accumulation of these nanoparticles. We know that. But more importantly, we actually see the stable oscillation. So it's a stable oscillation through this treatment. And we observe after 35 minutes later, which is in the regular bubble, everything will be already disappeared. But if they are the bubbles intact in the extravascular space, we actually observe the second harmonic generation. So that means we actually determine the intact nanobubble acoustic in the extravascular space. So this is very exciting because now this nanobubble not only allow us to improve the drug delivery, but more importantly, generating this intact nanobubble beyond the vasculature can lead to new imaging application. For example, the high frequency nonlinear imaging or therapeutic opportunity like sonodyne therapy, because now the particle, the bubble will be generated with acoustic behavior in the beyond the vasculatures. So second example I want to give is this called J-aggregate porphysomes. Why we want interested in that? Because porphysome is a multifunctional, but they are not, doesn't have the intrinsic stimuli responsiveness. They are multifunctional, but they don't change. They have no phase change. So in order to creating that, we actually learned a lesson from the nature. It's called photosynthetic bacteria, because the yellow stone has a beautiful, these hot springs and beautiful color. There was many of these thermal resistant bacteria. One of them very exciting, it's called Rhodosaphiroides. It's a photosynthetic bacteria. If you look at the detail of this structure, you can see this is coming with light harvesting complex is actually contain a two different shape of the porphyrin. The same molecule has a two different shape. They're holding together by a protein matrix. When they are in like a fence format, they're 850 nanometers. When they are in the distributed out in the plane, they're 800 nanometers. So this exciton coupling is also called de-aggregation. So my student came up with the idea, what if we recapture this nature's magic of these protein matrix in a porphysome, in the liposome structures? This is what we see, because the lipophilic tail of the porphyrin interact with the protein should be very similar to the lipid tail interact with the phospholipid. So indeed, when we actually introducing this bacterial lipid, will enable this into the liposome, we enable J-aggregation formation. That means a high ordered formation of this porphyrin. And this is called ordered aggregate, and lead to a very clear J peak, that is a 75 nanometer redshift peak, and which is basically consistent with the secondary structure, showing this CD spectrum. And with that, we're able to show these two phases, 750, 825, can be manipulated as almost like computer binary code, 1, 0, as a way to do the stimuli-responsive work. For example, if we introduce into the lipid, because lipid has transition temperature, if you increasing the temperature to above the transition temperature, these aggregate will be dissociated, even though the intact structure remain. So that means as soon as you see the aggregate disagreed, you actually see the blue shift, 75 nanometer blue shift. And this can be reversible, and it can be, this when we put in the PCR machine, we can see the 824 is green color, 750 heat it up, temperature, the wavelengths drop, you go back, you can keep doing this. So the reason why we're doing this, because we want creating a sensing mechanism, a stimuli sensing mechanism intrinsically. Here we see by using the green is our porphysome signals, red is a is ICG signal, does not associate with temperature. The idea is as a set temperature, in this case 45 degrees, you will see the green signal will vanish, right? Indicating, okay, here, this hitting the temperature, green signal disappear. It tells me the temperature is raised. I can use this as a thermal monitoring tool for the high field, for radio frequency, for photothermal ablations. More importantly, this is also providing opportunity for smart photothermal therapy, because the photothermal therapy has two limitations. One, once you shine the light, the temperature keeps going up. It doesn't stop. You're overheating. Second, when the light absorbs, they don't go further. So superficial heating. But because we have this built-in mechanism of thermal sensor, everything changed, because the light goes in, heat up. As soon as you're reaching the temperature, no longer absorb this wavelength because disaggregate. Now the light can go further and temperature will never overheat. So demonstrate this concept, we demonstrate. This is the control. You can see the length of the light goes through very clearly. if you're adding the porphysome gold nanoparticle or any other non-stimuli responsive way, you see the temperature can keep going up beyond our set temperature, but light cannot go through entire length. But if you're in the porphysome, J-porphysome case, you can see the temperature goes to certain degree, then stop. The light will penetrate through the entire length. And this we will see. This is what we call pearl, essentially a self-regulating porphysome. You see the light can go through, very clearly the entire length. Essentially, you're crossing the wall slowly, a person crossing the wall slowly, right? This is how we think we'll overcome the current photothermal ablation challenge. I think I have 5 more minutes. I can give you another example. This is about radiopharmaceuticals. As I mentioned, porphysome can chelating radioisotope like copper-64 for PET imaging. But porphyrin as a molecule, chelator, is not a very good chelator for many of these very exciting radiopharmaceuticals like lutetium, like these yttrium, like the terbium and all these. The reason is because the porphyrin has a ring is not big enough to holding this mostly as a 2+ very stable cationic. But if you have a 3+ ion, it's very difficult to keep them in the pocket. The one way to address that is to expand this porphyrin structure to expand the porphyrin from porphyrin to taxophrin, right? This is called taxophrin. In this particular case, you have a 4 coordination, a 4 nitrogen coordination of the metal becomes a 5, right? Plus exo 2, now you have a 7 key coordination site. Second, the pocket of this one going 20% bigger. Then this allow you to incorporate dramatically wide range of this, especially the lanthanide. Most of them are very useful for the radio theranostic for radiopharmaceuticals. So we have my student Joe demonstrated at that time we can actually using all these metal can be stably creating 1-to-1 ratio of these taxophrin into a nano taxophrin, which is taxophrin porphysomes. And now we're actually adding a lot more metal to this periodic tables. In fact, we were able to show by using the 2 metal as example, we actually can titrating these, forming this customizable, essentially making the taxophrin with the 2 metal confirm their ratio and confirm their quantitation of this metal chelation. So that means once we establish that, we can actually open up floodgate for different radiopharmaceutical application. This is where my student Miffy came up. For example, she actually did this one with the PMSA-targeted with a 2-3 modality using the cold lutetium 175 as a photodynamic therapy agent, using indium 111 as spec imaging agent, and the PSMA ligand for the PSMA targeting, right? So she, in order to do the radio label, she and the student Yulin in the lab creating this in-house microfluidic chamber. Essentially, because of the radioactivity, you want to make this one-time use, throw away, basically these cheap microfluidic chambers, so we can actually do the radio labeling very nicely, getting high radio labeling yield based on the radio ITLC, by the size occlusion, by the autoradiographic, and by the size measurement. So this is a very high yield. Once you do that, she was able to creating this nanotaxophrin with indium 111 for spec imaging showing the PSMA positive tumor versus negative tumor. You were able to show this cold lutetium, give a very nice fluorescence imaging of the PSMA positive tumors. You can show the photodynamic efficacy of this cold lutetium of the nanotaxophrin. You can also do the receptor targeting, right, by the PSMA with or without. So this, I hope, give you a flavor of what this can come. The nanotaxophrin, really the power of metal chelation is unlimited. For example, I want to guide you to this box. You can have different alpha emitters like lead 2212, bismuth 213. You can have a beta emitters, yttrium 90, lutetium 177, terbium 161. You have OG electron like thallium 201, not just the indium 111. Essentially, many of the radioisotopes we can take advantage of. for all these different applications. So it's glorified porphysome, but much more broadly. So that's essentially is my talk today. And I will need to thank a lot of people, particularly my first batch of graduate students is called Gangsters. And Zheng, who actually initially discovered this porphysome together with Chen and the Liz. You have the Chen did all this lung cancer work. Tracy did the established the radio labeling protocol. Ken did the stimuli response work. Liz did the microbubble work. And Joe and Miffy did the taxophene work. Carly did the nanobubble work. Michael, who actually really driving this translation pathway for porphysome, together with a lot of clinical collaborators, key people listed here is Dr. Wilson, Irish, Yasufuku, and Stephanie. So, and also grant support, including the Nanomedicine Innovation Network, who sponsors the tax-free project. Thank you very much. Happy to answer any questions.
Well, Dr. Zheng and his gangster team, thank you so much for this exciting research presentation. What a whirlwind tour of many, many years of exciting discovery and application and translational science. So,
well, I have to say that mostly, you know, we learned a lot of painful lessons. Hopefully, the next time, you know, will be much smoother.
well, you know something, I think those, you know, falling down, skinning your knees, the important thing is you get back up and you do learn those lessons along the way, and they inform both you, and hopefully you share them widely with your students and colleagues so that as they progress through their science, those are mistakes they can avoid. They can make new mistakes and share the results with you. So, you know, I think that's a very important part of the process. And
yeah, fantastic talk. That was very, very impressive. As a medicinal inorganic chemist, one of my interests is what different properties of the porphysomes do you get if you put in different metal ions? So do you get the same size and charge distribution, say if you stick indium in versus if you stick manganese or iron? What kind of variations are there in the properties of the porphysomes themselves as a function of the different smaller metal ions that you've been using?
So great question. Most of the metal we worked with using the nanotaxophene, essentially using expanded porphyrin because of the chelation requirement. But we observe in the porphysome case, when we insert copper 64 or insert manganese, has no change in terms of the morphology, the shape, the size of. There's no change, no influence on the nanostructures formation. Initially, we were worried when we're doing the taxophene because of the axial ligand, right? We worried about the axial, when you have axial ligand, will that impact the structure formation? We didn't observe any of that. So maybe because of the cholesterol, because of the other component of the bilayer, it mitigating the impact.
Okay, thank you very much.
Thank you.
Well, thanks for that question, Chris. So, Gang,
yes,
if you compare the commercial potential, the clinical impact potential of these nanotaxophene particles compared to lipid nanoparticles, what would you say the strengths and weaknesses of each? Are they complementary to each other in terms of the opportunities for treatment, or are they quite different and distinct in terms of their strengths and in terms of the application in the clinical setting?
So first is weakness. I like to point out the first. In terms of radiopharmaceuticals, radiotheranostics, most of them are still small molecule or the peptide based. There are reasons for that because accumulation of the nanoparticle in the liver and spleen is not ideal for the internal being essentially the radiopharmaceuticals. And that's why most approved ones are the smaller molecules, right? But nanoparticle has its advantage as well. I think particularly in terms of external beam radiation enhancement. So we are working on both because of the taxophene molecule itself is a very nice chelator. For example, thallium is 201 is a very, very nice OJ electron, but they are not, there's not a single chelator which really works well to work with compare with the thallium 201. And we observed the taxophene itself is a very good chelator for the thallium 201, which is a collaboration with the King's College and as well as the Ray Riley group. So the taxophene as a nanoparticle you can bring in the nano advantage, but don't underestimate the taxophene itself as a good chelator. So we were working on both small molecule and the large molecules. But another thing is, they are lipid nanoparticles. I mean, never doubt of that, right? So that's why we're working on using, using this external stimuli to trigger the cytosolic release enhancement of the RNA, right? Particularly because we know that's the next big things, LMP, and LMP has only limited the cytosolic release efficiency. So I think with our new function, bring either the light or X-ray or ultrasound, a different way we can, radiation, we can bring in, we will hope we can find a way to improve RNA delivery as a controlled mechanism. So that's what we're working on. Very exciting field.
Okay, gang, one last question before we wrap up. I think it was in association with some collaborations you were doing with surgeons at Sunnybrook. Did I hear you say that you were able to get the particles across the blood-brain barrier?
So that is with the Sunnybrook ultrasound group. Clover Hannaning, Dave Gertz, we have many years of collaboration because we can introducing the porphyrin into the microbubble.
Okay.
So that's called porphyrin microbubble. So that allow us have additional control and also have an in vivo imaging tracking tool to see what happened when the bubble was excited by ultrasound. And we were able to take advantage of that for a new imaging and the therapeutic applications.
Well, I think the takeaway here is there are so many incredibly important applications for your research that we all encourage you to continue and increase the number of gangsters working on all of these projects that you've got underway. So